Thank you. And probably this is the last talk that I have to substitute somebody for. So this was a study that we planned because we are used to use point-of-care analyzers in our practice. But one of the things of point-of-care analyzers is that they provide fast results, so it's usually faster than sent to a laboratory. They can be very cheap for something, or can be expensive for other things, so this cheap thing is a bit controversial. They are for sure easy to use, so you don't need a technician that run the point-of-care analyzer. They can be used, some of them can be used in field conditions. There are point-of-care analyzers that are really portable, like this one or this blood glucose meter. Some other point-of-care analyzers, like the VetScan, that requires like electricity, so can be used in field condition, but you need to arrange the way you are using it. And they usually require a lot less blood than normal laboratory testing. So this can be very important. We always work with, we usually work with small animals, with small turtles, small tortoises, small snakes. We don't want to have, we don't want to draw like 3 mL of blood from a 500-gram turtle. And so these instruments can be really useful in real life. One of the things that I think it's always important to realize is what are we really measuring? Are results accurate from these kind of point-of-care analyzers? And for accurate results, I mean, there are a couple of things that make accuracy, and one is trueness, and the other one is precision. As you may see here, all the values, let's say, are all together. This is something very precise, but it's not very true, let's say, because you want all the point here. And this other target show a point-of-care instrument that has good trueness, because all the results are quite near to the value that you really want, but it's not really precise. So you have results that are quite different from each other. We should imagine that these are all samples from the same animal, okay? And so the precision is also the repeatability of the measurement. So when you take 3 or 4 samples from the same animal and you analyze in the same time with the same instrument, how much they will get far from them. We had another POC instrument, point-of-care instrument, that we analyzed a couple of years ago. Was the iStat analyzer. Is a blood gas and electrolyte analyzer, and it had some clinically relevant disagreement, and so we checked what is out there about the VetScan in other chelonians, and there are definitely some interesting studies. There is one study on sea turtles, and another one in which they found acceptable results. It's always difficult when we do this kind of agreement studies, everyone has different level of acceptability, so it's very difficult to compare studies between each other. But in a study that included several reptiles, they found some differences between the reference analyzer and the VetScan. And this one is a very interesting study. It's an extensive review on all the point-of-care instruments that have been ever used in non-companion animals. So you should definitely have a look at this paper. But this paper is not about VetScan, because they say that it didn't fit their idea of portability. That makes some sense. There are some values for biochemistry of Hermann's tortoise. Like all the reptiles, there are differences between sexes, differences between seasons, but there is no data evaluating the performance of this point-of-care analyzer, so the VetScan in these animals. So the objective of the present study is to evaluate the performance of this VetScan in venous samples collected from Hermann's tortoises. And what we hoped, our specific hypothesis, is that the instrument had a good agreement, and so they could be used interchangeability. can be used one and the other without big differences, let's say. So this is a method comparison studies. So is a study in which we get one sample and we analyze with two different techniques, and we see which kind of differences there are. The primary outcome was the agreement between the instruments. And the animals were Hermann's tortoises. These are let's say, terrestrial tortoises, more common in I can say in Italy, but for sure also in Spain and in most of the Mediterranean area. Are kind of small, so medium size to small. So the largest female are 2 kilos, 2.5 kilos, but they're very big, or otherwise are around 1 kilo. And so we can't get like a lot of blood from this animal. And so they were clinically healthy, living outdoor, didn't have any significant issue. But what we cared about was the way they were fed with herbs, with healthy grass and everything. These animals were used for pharmacokinetic study, and so before the pharmacokinetic study, we had the opportunity to get some blood sample for having this biochemistry, and we think to do this opportunistic study on this instrument. So the benchtop analyzer is the VetScan VS2. Probably some of you have this instrument in your practice. And it can do a full biochemistry panel with 0.1 ml of blood, full in the sense that it's like 12 analytes, and there are interesting analytes, including like uric acid that can be useful in reptiles. There is battery acid, and so it's kind of complete for having reptiles, and takes around 10-15 minutes. The laboratory analyzer was the Olympus from the one department in the university, and so we obtain all the blood sample from the jugular vein. We try to always use this blood vessel because there were reported some troubles with the Charcinis reported 5 years ago interesting case series with the issues of doing subpartial sampling. So we try to avoid that site. And we used 1 cc syringes with 23 gauge needle, and we placed the blood in the lithium heparin tube. Immediately, before like 20 seconds, we run the VetScan. Then we centrifugate the sample, and we ship the sample for analysis with the laboratory analyzer. For the albumin, as also this laboratory analyzer use the bromocresol green. That is not ideal way to measure albumin in reptiles, or also in rabbits is kind of criticized. So for albumin, we performed protein electrophoresis. We used typical statistical analysis for these Bland-Altman plots plus Simba block regression. And if we had missing data, for example, if we had a system failure for a single or multiple value, the results of the remaining results would have been included in the study in any way. We didn't have missing values in the study, luckily. Were 50 males, 8 females, 550 grams to 2 kilos, 0.3. And these are the main findings. So we had underestimation of the albumin, underestimation of the AST, proportional bias for calcium, and what else? For glucose was overestimated by the point-of-care analyzer. Phosphorus was overestimated. Potassium underestimated, and total proteins underestimated. I mean, this is not really, it's a bit difficult to go inside every one of the analytes, but just quickly, for example, aspartate aminotransferase, you can see that the differences between the point-of-care analyzer and the reference analyzer are somehow not so big for an enzyme that raise like of thousands. So for ST, it could be interesting, this VET scan. But for example, the creatine kinase also had a very wide limit of agreement. So there was a difference, a median difference of -10, but from 123 to 140. So if you take a look at this value, for example, the lab analyzer measured 250 units per liter, and the VET scan measured less than 35. So, I mean, creatine kinase is not one of the most important analytes that we evaluate, but the blood glucose was kind of interesting because there was a significant overestimation of blood glucose. Here, it cannot be properly seen, but there is the real, let's say, the zero difference line, and all the results are moved up here. So this means that, for example, a value of 30 from the VET scan corresponded to a value of 20 from the reference analyzer, a value, and so on. So the mean difference was 12.8 milligram per deciliter for the glucose. And this was also found in other species, like in the Caretta caretta, there was a mean overestimation of 5 milligram per deciliter. And in other reptiles, there was a mean overestimation of 10 milligram per deciliter. So there seems to be, there has to be something there that make difficult to measuring glucose in reptile blood. Because otherwise the VET scan work properly. For example, in rabbits, it worked perfectly for the glucose, so this is very strange. So the uric acid, for example, had several values that were very different from the reference analyzer. This one is 4.5, and the reference analyzer is 3. But everything was inside like the reference interval of the turtles, of this tortoise, so probably not clinically significant, just a difference in measurement. And albumin, like in other studies, they have proved a few years ago that the bromocresol green dye binding method is not accurate for measuring albumin in turtle, and our study had the same results. So yeah, protein electrophoresis should be always used when somebody suspects like a decrease in albumin or something like that. So there are some limitations of this study. We included only healthy animals. That usually is something good for other kind of studies, but for this type of study is not good because you have a narrow range of all the analytes. It was a small sample where less than the 30, 40 animals that are suggested for method comparison studies. And this was because obviously there were some financial and time limitations. I mean, and these were the animals that we had at that time. And obviously we used plasma and not serum because seration is sometimes difficult to predict in reptiles. So we only evaluated trueness, and we didn't evaluate the precision, so we didn't have multiple samples analyzed with the VET scan, and this is a further limitation. And for concluding, I want just to think about, are fast results always better? It's unclear. In human medicine, they have different studies in which they prove that the use of point-of-care analyzer may fasten the diagnosis and may ameliorate the diagnosis, but sometimes they have other studies that say that there are different clinical decisions with point-of-care analyzer, so we should use this instrument very carefully. In conclusion, the uric acid has an acceptable agreement for our clinical point of view. For the glucose, we can use the rule of thumb of removing 10 milligram per deciliter from the VET scan results to have the laboratory result. And for albumin, we suggest to use protein electrophoresis. For the other analytes that have lack of interchangeability, there is the need of instrument-specific reference ranges, and the use of the same instrument is suggested to monitor the same animal. Okay, I want to thank you, Dr. Giordano Nardini, Paolo Selleri, and Gloria Isani, that we made this work together, and thank you for being here.